Hi students, welcome back. Welcome to the last session of Pharmaceutical and Medicinal Chemistry Terminology students. In this last session, let us look into drug development and monograph of drugs, which is a very interesting topic students. Let us learn about the drug development. Okay students, let us learn about the drug development. Drug development means how was the drug invented and what are the types of that drug, how was that invention was carried forward. All these things we are going to learn in this video students, which is very interesting students. For this drug development, I have chosen example of antibiotic penicillin students. uh, Antibiotics are nothing but these are the drugs which are prepared from useful microorganism to kill or to invade the disease which which were caused or which were created by a harmful microorganisms. The drug itself was prepared from a microorganism to kill another microorganism. The one line of matter is the drug is prepared from a useful microorganism to kill harmful microorganism. So for this drug development, I have chosen example of penicillin students. Right. Penicillin is an antibiotic students which is produced by one microorganism that is toxic to kill another microorganism. So this penicillin discovery was an accident students. This accidentally it was discovered students. Okay. Earlier stories says that the scientist kept away the species of bacteria, some culture. He has grown some culture of bacteria and he kept that grown culture near the window and went for a vacation. After he returned from vacation, he observed that that culture was spoiled. So he did further investigations what happened to spoil that grown culture. So finally he concluded that another microorganism which is useful microorganism killed that culture of bacteria microorganism which is harmful, which is nothing but Penicillium notatum species. So it was said as a story students. Okay. It finally penicillin was discovered accidentally students. The accidental discovery of penicillin, one of the most widely used antibiotics in the world students. It was observed by Alexander Fleming in 1920s, right? This discovery was done by or Alexander Fleming in 1920th century itself, that bacteria does not flourish on nutrient agar in the region surrounding particular fungus Penicillium notatum. He suggested that the fungus produces an antibiotic named as penicillin, right? What happened in 1938, Ernest Chain and Howard Florey isolated penicillin in pure form and proved its effectiveness as an antibiotic. It was introduced into medical practice only in 1941. Such a huge gap student, it was invented by Alexander Fleming in 1920, but it was came into medical practice in 1941. Since then, several naturally occurring penicillins have been isolated. All these penicillins have empirical formula, which is very important students. That is nothing but C9H11O4SN2R. You have to remember it. No other way. So all the penicillin have same formula. Okay, penicillin is an antibiotic. Okay, it is prepared from Penicillium notatum species, right? And the molecular formula of penicillin is C9H11O4SN2R. Okay students, in penicillin, four-membered ring. Let me show you. In penicillin, the four-membered ring is B, is fused to another five-membered ring which is A. B is a four-membered ring students, and A is a 5-membered ring. This 4-membered ring is clubbed or fused to a 5-membered ring. This is the structure of all penicillins. Common structure of penicillin contains a 4-membered ring clubbed with a 5-membered ring. Okay students, a number of penicillins differing only in the nature of R group. If you observe here, they differ only in the nature of R group. The remaining nucleus will be same. The remaining nucleus all will be same. They differ only in the R group. They differ only in the R group. Remaining 4-membered ring and the 5-membered ring will be same. Only the R group will be differed. Okay. R group have been isolated from natural media, right? And more than 100 have been synthesized. Penicillin G. Many have been synthesized, students, but penicillin G is the most commonly used penicillin derivative. So, the general structure of penicillin is like this. Okay? A B, a 4-membered ring, 4-membered ring with a 5-membered ring with a R group. Okay, this is the general structure of penicillin. The R group will be replaced. So, by replacing the R group, we'll get a lot of penicillins combination. So, let us look into forward this. So, what is the A ring, students? Let me keep like this so you can understand. A ring is thiazolidine ring and B ring is beta-lactam ring, right? If in R group, in the R group, if you place CH2, CH double bond CH, CH2, CH2, okay, that is pent-2-enyl penicillin, which is nothing but penicillin 1 or F. This is called as penicillin F. If you place in the R group, CH2 double bond, CH2, CH double bond, CH, CH2, CH2, which is pent-2-enyl penicillin, this is called penicillin 1 or F. If in the R group, if you place CH2C6H5, if you place CH2C6H5, then it is called as benzyl penicillin, that is penicillin 2 or G. If in the R group, if you place CH2C6H4OH, that is para-hydroxybenzyl penicillin, it is called penicillin 3 or penicillin X. Okay. Next, if in the R group, if you place CH2 taken 6 times CH3, then it is called N-heptyl penicillin, which is called as penicillin 4 or K. Next, if in the R group, if you place CH2O C6H5, which is called as penicillin V, that is CH2O C6H5 group means phenoxymethyl penicillin. Phenoxymethyl penicillin, then it is called penicillin V. Next, if in the R group, if you place CH6H4 OH NH2, that is alpha amino para hydroxypenicillin, then it is called as amoxicillin. Next, if in the R group, if you place CH6H5 NH2 amino benzyl penicillin, this is called alpha amino benzyl penicillin, which is called ampicillin. These are the types of penicillin, students. This table can be very important, and it can be also be studied under the heading classification of penicillins. This also can be studied under the heading of classification of penicillin. This is about how the penicillin was discovered and how many types of penicillins are there by replacing R group. Next, isolation of penicillin. How the penicillin notatum species is isolated, right? First method is submerged cultured method. Okay, penicillin on a large scale can be produced by various culture methods. The main method which is used for commercial production of penicillin is submerged culture method, is a commercial production of penicillin. In this process, what we are going to do is Penicillium chrysogenum is used for the culture. The culture medium used for this 1000 parts contains the following ingredients. These following ingredients are used for submerged cultured method. Okay, they are lactose 20 parts, corn steep solids 20 parts, sodium nitrate 3 parts, dipotassium phosphate 0.05 parts, magnesium phosphate 0.012 parts, calcium carbonate 1.8 parts, phenyl acetic acid 0.5 parts. These are all the ingredients which are used to grow the penicillin in the submerged cultured method, right? In this method, the culture and culture medium are taken in large tanks and the medium is agitated by a steam sterile and the temperature should be maintained at 24 degrees for 2 to 3 days. So we will take all these things in large tanks, okay, and the temperature should be 24 degrees and this is done for 3 days. After this fermentation in this vessel, the vessel will be sealed and small positive air pressure is maintained inside the vessel. So the harmful material growth for the atmospheric air may not enter the vessel. So under these conditions of agitation and aeration, continuous agitation and of aeration, the mold grows through the bulk of liquid as a globular pellicles consisting mainly of mycelium. Okay. So what happens? How the penicillin is isolated? This growing of Penicillium notatum species, growing penicillin in large tanks, fermentation taking place for 3 days and the temperature for 24 degrees centigrade, everything will be okay. The mold will be grown, the species will be grown. But what happens? To isolate the penicillin, what are the problems we are going to face in submerged cultured method? Penicillin is readily decomposed by 2 acidic or by 2 alkaline solution and it is also decomposed in heavy metal ions. Penicillin is destroyed. If we raise temperature shortly, the grown pencil for 3 days will become useless. It will be destroyed. So thus complete aseptic conditions during penicillin production should be maintained. So these will be problems in isolating of penicillin. So what were the steps has to be taken to separate penicillin, to isolate penicillin? After fermentation, the content of vessels are rapidly cooled and the mycelium is filtered on a sterile rotatory filter. First we have to cool those things after fermentation and we have to filter. Next step, we have to adjust the pH of the filtrate. So the magnitude of pH depends upon the nature of solvent of extraction. If amyl alcohol is used as an extracting solvent, the pH is adjusted to 2 to 3. So it depends upon the solvent we are using for extraction. If we use amyl alcohol, pH should be 2 to 3. Butyl alcohol, it is adjusted to 6.4 by adding ammonium sulfate. After adjusting the suitable pH of the filtrate, solvent extraction either with amyl alcohol or butyl alcohol is carried out. Okay, after if you adjust the suitable pH of the filtrate, solvent extraction either with amyl alcohol or butyl alcohol is carried out. Now the petroleum ether is added to the cold solvent extract and this is shaken with sodium bicarbonate solution. First we have to take all those things. What all those things? We have to take lactose, corn steep solids, sodium nitrates, dipotassium phosphate, magnesium phosphate, calcium carbonate, phenyl acetic acid. All these things we have to take in large tanks and the temperature is maintained at 24 degrees and we grow in large tanks. And next what we will see? We will extract with the isobutyl alcohol or amyl alcohol. After that we will adjust the pH, right? After that pH is adjusted, we will cool the liquid. After that we will add petroleum ether and again we will dilute with sodium bicarbonate, right? Now pH will be adjusted to 6 to 7 and then it is rapidly frozen and evaporated to yield the sodium salt. To set pure sample, it should be dried in high vacuum. We have to dry it in high vacuum. The penicillin is generally administered by injections. We know that penicillin injection is first tested. It has given very light to our skin. They will mark a round mark with pen, and with that, in that area they used to inject penicillin. And if the doctor used to observe if any irritation or any side effects are present, they stop giving that penicillin. If no side effects are observed in that particular area which is marked and injected, if it is clear, crystal clear, then they will inject to our body. So the penicillin is generally administered by injections. The final sample must be non-toxic, sterilized and free from pyrogens, right? The penicillin obtained from the above methods is not one penicillin but mixture of number of penicillins. Okay. However, if a suitable chemical compound is added to culture medium, it becomes possible to increase the yield of the desired penicillin, as well as it becomes possible to produce new types of penicillin. If penicillin is administered orally, the stomach juices possesses the enzyme penicillinase, which also hydrolyzes penicillin, opening the lactam ring with loss of activity. So, if it is not injected, if the penicillin is not injected, if the penicillin is taken orally, what happens in our stomach? Penicillium hydrolase enzymes are present. They open our lactam ring, which is the reason for activity. That lactam ring is particularly the reason for antibiotical activity. If that ring is open, what is the use of taking penicillin? So that's why penicillins loss activity if they are taken orally. That's why they are taken in the injection form. Penicillin G, benzyl penicillin, is considered to be the most effective antibiotic for the treatment of greater variety of bacterial infections than any other antibiotic, except for the persons who are allergic to penicillin. Oh, lot of patients use antibiotic as penicillin, except for those persons who are allergic to penicillin. Okay. We have already seen in the previous table. So in this previous table, what happened? I have shown the R group here, and I told in the R place you can produce substitute these groups. Now let us write the structures of few penicillins by substituting R group. See, if you observe, this was the R group, right? In the R place, I have substituted C6H5CH2, which is nothing but penicillin G. And in R group, here in the R group, I have substituted C6H5OCH2, which is called penicillin V. And in here, in this R group, I have substituted this part. CHNH3COOH-CH2 taken 3 times, which is called as penicillin N, which is also known as cephalosporins. And in the R group, if you substitute this group, benzene with CH2, NH2 and OH, amino para-hydroxybenzyl penicillin, then it is called as amoxilin. Okay, these are few different types of penicillins. What is penicillin G? What it is used, how it is used. Penicillin V, what for it is used. Okay, let us learn in deep. Penicillin G is called as benzyl penicillin. It is a natural penicillin and most effective antibiotic for the treatment of greater variety of bacterial infections. I mean, it is active against some kind of bacteria, for example, pneumococcus, streptococcus, gonococcus, streptococcus, anthrax, clostridia, penicillinosis negative, staphylococcus, and against actinomyces fungus. All this is active against all these kind of bacterial infections. What about penicillin V? Benzyl penicillin is penicillin G. Phenoxymethyl penicillin is our penicillin V. It is used for treating mild infections of respiratory tract produced by gram-positive organisms such as streptococci and pneumococci. It is very useful in prophylaxis of rheumatic fever. We use in rheumatic fevers penicillin V. So the penicillin V is used to treat respiratory infections, mild respiratory infections, which are caused by gram-positive organisms such as streptococci and pneumococci. Okay. And what is penicillin N? Penicillin N is also known as cephalosporins. It is 4-amino-4-carboxybutyl penicillin. It is less effective against gram-positive bacteria. Penicillin V treats mild effects which are produced by gram-positive organism. Cephalosporins is also used, but it is less effective against gram-positive bacteria. Amoxilin. Nowadays, many of us use amoxicillin, which is a famous antibiotic, which is prescribed antibiotic, and worldwide it is used in very large doses, right? Amoxicillin. It is an antibiotic which is used for oral administration. So how amoxicillin is prepared? So that if you take orally also, when it enters into our stomach, it will not be hydrolyzed and the beta lactam ring will not open up. So in the starting investigations, only injections has been prescribed, but nowadays it has been taken orally also, for example amoxicillin. Now let us move into the another interesting topic, drug monograph. Drug monographs gives us the full information of one drug, about its texture, how it is used, what is the dosage of it, what is the source of supply, how it is called as, what names are used, what is the generic name, what is the chemical name, what is its solubility. All those things come under drug monograph. Every drug needs a monograph. If you know the monograph of every drug, we can easily prescribe the medicine for a disease. Of course, we have to consult our physician. Without physician or doctor's prescription, we should not take that medicine. But as educated students, we should know the monographs of drugs, so that we know that what happens when a drug is taken into body, how its function, how it is excreted. All those things we can learn in the monograph of drugs. Having knowledge of medicines, what we are taking is a good habit, and we as a chemistry students, we should know why we are using that drug, what is the chemical structure of that drug, how it is used, what is the therapeutic activity of that drug. Okay, it is the statement that specifies the kinds and amounts of ingredients of a drug or a class of drugs may contain, the directions for the drugs use and the conditions in which may be used. Okay, a monograph includes the name of the ingredient or preparation, packing, storage, labeling requirements, and the specifications, right? The specifications consist of a series of tests, procedures for the tests, and acceptance criteria. Okay, these tests and procedures require the use of official USP. Okay, we should follow all these things so that we will know that whether we are taking a right drug or not. Is that drug is of standard or not. Okay, reference standard. Medicinal ingredients and products will have the stipulated strength, quality, and purity if they conform to the requirement of the monograph. So every drug will have a monograph, and we have to know that monograph. For example, paracetamol, which is a wide drug which is used for reducing body temperature, which is used as antipyretic to reduce our body temperature. Okay, we are taking Dolo 650, right? Kids take P250, P500, etc. So these are all comes under paracetamol. The medicine is nothing but paracetamol. What is the generic name of this? Acetaminophen. What is the generic name of that paracetamol? Acetaminophen. Okay, what is the source of supply, trade name and manufacturer? Paracetamol, right? Physical properties of paracetamol. What is the structure? Benzene ring with OH and NHCOCH3, which is quite opposite. Benzene ring with OH, 2 OH para position, NHCOCH3 will be present. That is the structural formula of paracetamol. What is the molecular formula? C8H9NO2. A. Structural formula. B. Molecular formula. Molecular weight is 151.16. What is its appearance? It is a white crystalline powder, right? What is its solubility? At normal temperature, 1 part of it dissolves in 70 parts of water. In boiling water, 1 part it dissolves in 20 parts of water. Solubility in other solvents is given below. Alcohol 1:10, chloroform 1:50, glycerin 1:40, either slightly soluble. This is about the solubility of paracetamol. What are the chemical properties of paracetamol, right? Structural similarities, differences of a drug to other available compounds or group of compounds. Paracetamol, which is known as acetaminophen, is a synthetic non-opiate centrally acting analgesic derived from para-aminophenol. It is an antipyretic as well as analgesics. It is a painkiller also, right? The full chemical name is N-acetyl para-aminophenol. What is its acidity constant? Is 9.51 at 25 degrees C. Is the drug is stability to the drug temperature, light and moisture? Yes, it is stable to temperature, light and moisture. What is the range over pH? Which drug is stable in solution? Acetaminophenol is stable at pH between 4 and 7 at 25 degrees. And acetaminophenol oral solution, adult liquid, has a pH of 3.8 to 6.1, and the oral suspension for infants, drops for children, is pH of 5.4 to 6.9. If acetaminophenol oral solution is applied to adults, the pH should be between 3.8 to 6.1. And if it is prescribed for infants, I mean small kids, the pH should be between 5.4 to 6.9. Okay. Extra strength Tylenol acetaminophen adult liquid 3058 plus or minus 150 millimole per kg. Okay, these are all the prescriptions for adults, and for children's it will be as Tylenol osmolality complex. At the R acetaminophen elixir 60/40 plus or minus 25 millimole per kg. These are all the things which come under monograph of a drug. Okay students, in this class we learned a very interesting topic, which is nothing but drug development. In this drug development, we have come across the development of penicillin. How the penicillin was accidentally discovered by Alexander Fleming, and in 1941 it came into existence as medicinal value, and it has two rings, A and B. A is a 5-membered ring, B is a 4-membered lactam ring, which due to the activity of penicillin is due to beta lactam ring. I have also discussed about the types of penicillins: penicillin F, penicillin G, penicillin X, penicillin K, penicillin V, amoxicillin and ampicillin. I have also discussed about the isolation of penicillin. What are the contents? What are the 100 parts we have to take in, and we have to maintain it for 24 degrees. We have to take in large tanks. We have to take it in large tanks for 3 days. Okay, all these things, isolation of penicillin, and we have also learnt about the structures of penicillin, which are very important. Penicillin G, penicillin V. Penicillin G is used against pneumococcus, streptococcus, gonococcus bacteria. Penicillin V is used for rheumatic fever, and penicillin N, cephalosporins, is less effective against gram-positive bacteria. And we have also learnt not only penicillin is taken as in injection forms, the penicillin was in advance was changed as amoxicillin, and these are also taken as orally administered into our body. This, and finally we have learnt also about the drug monograph. Drug monograph is nothing but the complete information. The complete information of the drug is present in the drug monograph. For example, you can study any monograph, students, but for example, I have taken the example of paracetamol, which is an analgesic, okay, which is used for reduction of body temperature. I hope students, these are all the 5 videos of pharmaceutical terminology in very depth, students. Okay, you have to remember the each and every heading and learn the structure of drugs, which is very useful for your information personally, which is used in your lives, and also you have to write in your examination point of view. So this first unit completely contains what is a drug, what is a disease, what are the drugs used, what is the classification of drugs, what is a monograph of drug, how the drug is developed. All these come under your unit 1 students. I hope you understood this unit. Please make a short notes of it for your examinations, and all the best students. We'll meet in the next classes. Thank you very much.